So dear students, in the last class we were discussing regarding the anatomy of the plant. In the anatomy of the plant we have discussed regarding the different types of tissue involved in the stem, root and leaf. We have discussed generally. Now first we will start with the anatomy of a dicot stem. So dicot plant stem, when we take a section of a dicot stem, it appears in this view. So this is a view how if we take a TS and one section stem in section itara TS transverse section tagdaga itara spherical view kanutte. This is the anatomy of the dicot stem. The root the stem section, it looks in this form. When we focus into if we increase the magnification for a particular region in the microscope, so we will see this is the anatomy of a dicot stem. So the dicot stem, mainly it is classified into three regions. One is outer epidermis, second one is middle cortex, third one is inner stellar region or steel region. So these are the three different regions what we can distinguish in a dicot stem. So the outer layer, outer layer is the epidermal region. Next comes up to this yellow region, up to this region, that is the cortex. Then comes this entire region is a stellar region. So see, we have focused only to this region here. So entire stem, it is spherical, the section. We have taken only this part and we have focused under the microscope. In a compound microscope we have focused only for this particular region. Understood? So when we focus for that particular region, it appears in this form. So we can study the different layers. Now first we will take up with the outer epidermis. What is the epidermis made up of? In the dicot stem means the epidermis region, it is made up of barrel shaped parenchyma cells, which is compactly arranged with one another without any intercellular space. See, this is a single layer epidermis, the outer layer, single epidermal layer, and certain epidermal cells, they are modified to form a stem hair called as trichome. So I will write it here once again. So just I will write 4-5 cells. Understood? So this is outer epidermal layer. The epidermal layer, it is certain epidermal cells, they are modified to form the stem hair, or it is also called as trichome. It is called as trichome. But remember, the stem hair or trichome, it is multicellular. It is not unicellular. But in case of root hair, it is unicellular, whereas the stem hair, it is multicellular. So this is a stem hair. It is multicellular. Next, so this is about the epidermis. So next to the epidermis is the cortex. Next to epidermis is cortex. So the cortex, it is differentiated into three regions here. That is the outer cortex, epidermis and kedabhaga cortex. It is differentiated into three regions. Outer called as hypodermis, next middle general cortex, and inner endodermis. outer hypodermis, middle general cortex, and inner endodermis. So now below the epidermis, the 2 to 3 layer cells, it is called as hypodermis. The hypodermis, it is made up of 2 or 3 layers of colon-chyma tissue. What type of tissue is seen in hypodermis? That is a colon-chyma tissue. So you can see here, you can easily distinguish here in this. See, this is upper epidermis. Upper epidermis. Below the epidermis, you can see there is a thickening out around the cells. So you can see a thick layer of the cells. So that is due to the deposition of pectin. So we have already discussed the colon-chyma tissue. So the colon-chyma tissue, there is the cell wall, it is made up of it, or the cell wall, in the cell wall there is a deposition of pectin. We can see a pectinization in the colon-chyma tissue. So the hypodermis, here it is made up of colon-chyma tissue. Next to the hypodermis is the general cortex. So see, you can differentiate. This is the 2 or 3 layer cells, it is a hypodermis. Next to the hypodermis, the 2 or 3 layer cells, this is a general cortex. See, the general cortex here, it has made up of parenchyma cells, which is loosely arranged. So you can see here the intercellular space in between the cells. So this layer, not above the yellow color. Whatever this yellow color line is there, above that you see, here the cells, they are loosely arranged, whereas above that cells, here it is a thick cell wall. So here it is colon-chyma. Next, general cortex is this layer. This general cortex, it is made up of loosely arranged parenchyma cells. Loosely arranged parenchyma cells. So hypodermis, you have to remember the names. Which region is made up of what type of cell? That is very important. So the epidermis outer, it is made up of single layer parenchyma, barrel shaped parenchyma cells without any intercellular space. Certain epidermal cells, it is modified to form a stem hair, or you can also say it as trichome. Next to that is the cortex layer. The cortex layer, it is differentiated into 3 regions: hypodermis, general cortex and endodermis. So the hypodermal region, it is made up of the 2 to 3 layer of colon-chyma tissue. Next comes the middle general cortex. The general cortex is this one, 2 to 3 layer. This general cortex, it is made up of loosely arranged parenchyma cells. Next, moving on to the endodermis. The endodermis, it is made up of, it is a single layer. It is made up of single layer. Whereas general cortex also, it is made up of 2 or 3 layer. Whereas here the inner endodermis, it is made up of single layer barrel shaped parenchyma cells, which is compactly arranged. And the endodermis, here there is a deposition of starch inside this cell. So the food, it is stored in the form of starch, and starch is a storage form of food in plants. So hence there is a deposition of large number of starch inside this endodermal cells. This is the endodermal cells. So hence it is also called as starch sheath. ಅದನ್ನ ಏನಂತ ಕರಿತೀವಿ? ಸ್ಟಾರ್ಚ್ ಶೀತ್ ಅಂತ ಕರಿತೀವಿ. ಯಾವುದು ಸ್ಟಾರ್ಚ್ ಶೀತ್ ಅಂದರೆ ಯೆಲ್ಲೋ ಕಲರ್ ಲೈನ್ ನೋಡ್ಬೋದು. ದಿಸ್ ಇಸ್ ಅ ಸಿಂಗಲ್ ಲೇಯರ್, ಎಂಡೋಡರ್ಮಲ್ ಲೇಯರ್. Understood? This single layer of a cells, parenchyma cells, which is compactly arranged. Single layer parenchyma cells, compact andre without any intercellular space. So this layer, the cells inside this layer, there is a rich, or there is a more number of starch deposited in this cells. Hence the endodermis is also called as starch sheath. So next to the endodermis is the stellar region. So we have studied with the epidermal region, and we have studied up to this yellow color. This is a cortex region. Now next we'll move on with the stellar region. Or steel region. The steel region is a centermost region, ಈ ರೀಜನ್. ಇಲ್ಲಿ ಇದು ಅಪ್ಪರ್ ಇದೆ ಎಪಿಡರ್ಮಿಸ್ ಆಯಿತು, ಇಲ್ಲಿ ಮಧ್ಯದಿಂದ ಇಲ್ಲಿಂದ ಇಲ್ಲಿವರೆಗೂ ಎಂಡೋಡರ್ಮಿಸು. ಈಗ ನೆಕ್ಸ್ಟು ಇಲ್ಲಿ ಒಳಗಡೆ ಈ ಒಳಭಾಗದೆಲ್ಲ ಏನು ಸ್ಟೀಲಾರ್ ರೀಜನ್. ಮತ್ತು ಈ ಸೆಂಟರ್ ರೀಜನ್ ಪಿತ್ ರೀಜನ್. ದಿಸ್ ಇಸ್ ಎಂಟೈರ್ ಸ್ಟೀಲಾರ್ ರೀಜನ್. ಇದಿಷ್ಟು ಸ್ಟೀಲಾರೇ. Now the stelar, it is differentiated into 3 tissues. The outer layer of the steel is called as pericycle. Outer layer of the stelar region is called pericycle. Next, middle layer or next comes the vascular bundles. Vascular bundles. And third, at the center, pith region, PITH. So see, there is a pericycle, next comes vascular bundles, and the centermost region is a pith region. So what is this pericycle layer made up of here antandre? The pericycle, so stelar region, it is differentiated into outer pericycle, vascular bundles and central pith. So the vascular bundles, you have to remember, it is made up of xylem and phloem tissue. Food and water conducting tissue are the tissue which is seen in a vascular bundle. Next, conducting tissue anta kuda kelbodu. Xylem and phloem is the answer. First we will take up with the pericycle. So the pericycle, in case of dicot stem, it is discontinuous. It is discontinuous. Why it is discontinuous means it is made up of a patch of sclerenchyma cells. Sclerenchyma. So sclerenchyma we have already discussed. It is a dead mechanical tissue. It is a simple permanent tissue, but it is dead. This is a sclerenchyma tissue. Next comes the parenchyma tissue. Next again comes a patch of cell which is made up of sclerenchyma. So when we'll write the sclerenchyma somewhat in pentagon or hexagonate shape, which shows the edges or which possess the corners. ಈ ಥರ ಬರೆಯೋದು, ಎಡ್ಜಸ್ ಇರಬೇಕು ಅಥವಾ ಕಾರ್ನರ್ಸ್ ಥರ ಇರಬೇಕು. ದಟ್ ಈಸ್ ಅ sclerenchyma tissue. So the sclerenchyma, you can see it is not continuous. In between the patch of sclerenchyma, we can see a parenchyma. So this is sclerenchyma. And this is parenchyma. This is sclerenchyma. Sclerenchyma. So see, the sclerenchyma, it is not continuous. In between you can see a parenchyma tissue. Hence it is discontinuous. And the sclerenchyma, it is present above the vascular bundle. It is present above the vascular bundle. Idu vascular bundle. ಈ ವ್ಯಾಸ್ಕ್ಯುಲಾರ್ ಬಂಡಲಿನ ಮೇಲ್ಭಾಗದಲ್ಲಿದೆ ಅದು ಯಾವುದು sclerenchyma. Hence it acts like a cap on the vascular bundle. ಯಾವುದು ಈ sclerenchyma tissue, it acts like a cap on the vascular bundle. vascular bundle ಮೇಲೆ ಒಂದು ಕ್ಯಾಪ್ ತರ ಕೂತಿದೆ. So hence this pericycle, which is made up of sclerenchyma, it is also called as bundle cap ಅಂತ ಕೂಡ ಹೇಳ್ತೀವಿ. What it is called? Bundle cap. Why it is called as bundle cap? Because it is present above the vascular bundle in the form of cap-like structure. Hence it is also named as bundle cap. Next to that is the, so this is about the pericycle. So the pericycle, it is discontinuous. Idalla vascular bundle barala pericycle ige, no. So just to explain the bundle cap, I have written the vascular region. the vascular, the pericycle, it is discontinuous because the sclerenchyma tissue, it is arranged in the form of patches. In between the two patch of sclerenchyma, we can see a parenchyma tissue. Hence, it is discontinuous. So, next is the vascular tissue. Next is vascular tissue. So the vascular tissue, it is made up of a xylem and phloem. So the xylem and phloem, first we will take up with the words. What type of vascular bundle it is? It is conjoint. We can say what is conjoint, we have already discussed in the last class. Conjoint andre eno? If the same bundle possesses both xylem and phloem, then it is called as conjoint. Conjoint andre xylem phloem ಒಂದೇ radius ಅಲ್ಲಿ ಇದೆ, ಒಂದೇ radius ಅಲ್ಲಿ ಇದೆ. It is conjoint or it is present together. Next, it is vascular bundle. It is collateral. Collateral. What is collateral ಅಂತಂದ್ರೆ xylemu ಸೆಂಟರ್ ಕಡೆ ಇದೆ, phloem ಅವೇ ಇದೆ. ಅರ್ಥ ಆಯ್ತಾ? xylem towards the center ಇರುತ್ತೆ, phloem ಅವೇ ಇರುತ್ತೆ, that is collateral. Next, it is open type. It is open type. So what is this open type of vascular bundle ಅಂತಂದ್ರೆ in between the xylem and the phloem, if there is a patch of cambium, if there is a patch of cambium cell. What is this cambium? Cambium is a meristematic tissue which is present in between the vascular tissue. Vascular tissue ಯಾವುದು? xylem ಮತ್ತು phloem. ಈ ಎರಡು vascular tissue in ಮಧ್ಯದಲ್ಲಿ meristematic tissue ಇರುತ್ತೆ. So we have studied in the first class when we have started with this chapter, anatomy of flowering plant. So the plant tissue, it is mainly classified into meristematic tissue and permanent tissues. So then the permanent tissue was classified into simple and complex. So here there is a patch of meristematic tissue. That meristematic tissue itself is called as what? Cambium. So this cambium meristematic tissue, if it is present between xylem and phloem, then that type of vascular bundle is called as open type. So then what is the function of this cambium andre? This cambium further it helps in the secondary growth of the dicot stem. Secondary growth agbekare in this chapter we are going to study the secondary growth in dicot stem. There this cambium will help. So hence if the cambium is present in between xylem and phloem, then it is called as open type. There is another type, closed type anta barutte. Ell barutte adu monocotyle barutte. Closed type vascular bundle ಅಂತಂದ್ರೆ the xylem and phloem in ಮಧ್ಯ cambium ಇರಲ್ಲ. If the cambium is absent, then it is closed type. If the cambium is present, then it is open type. So the open type, it is mainly seen in dicot stem. So hence it is conjoint, collateral, open, and it is endark. What is the word endark refers to andre? You can see here in the diagram. ಇಲ್ಲಿ ನೋಡಿ, ಇಲ್ಲಿ ಒಂದೇ ಒಂದು ವ್ಯಾಸ್ಕ್ಯುಲಾರ್ ಬಂಡಲ್ ಇಂದು ಫೋಕಸ್ ಮಾಡಿದ್ದೀವಿ. ಒಂದೇ ಒಂದು ವ್ಯಾಸ್ಕ್ಯುಲಾರ್ ಬಂಡಲ್ ಇದು. ಇಲ್ಲಿ ಮೇಲ್ ಪ್ಯಾಚ್ ಕಾಣಿಸ್ತಿದೆಯಲ್ಲ, ದಿಸ್ ಬ್ಲೂ ಕಲರ್ thick layer, this is what? This is a sclerenchyma, pericycle. Pericycle ಆದಮೇಲೆ ಈ ಗ್ರೀನ್ ಕಲರ್ ಇದೆಯಲ್ಲ, this is a phloem. Understood? This is a phloem tissue. Then to the phloem, this yellow color cell, these are the cambium, a patch of tissue, cambium. Next to cambium, this is a xylem. See here you can differentiate. The protoxylem is a smaller one, which is the first formed xylem. The later formed xylem with a with a wide lumen, it is called as metaxylem. This is proto and this is metaxylem. If the protoxylem is towards the center and metaxylem is away from the center or towards the pericycle anta kuda helbodu, ee pericycle kade. Idu towards the center. Then that type of vascular bundle or that type of a xylem is tissue, it is called as endar condition. Illu kuda helbodu nodi, illu gottagatte. Idu ಒಂದು ಸೆಕ್ಷನ್ ಇಂದು ಇಷ್ಟು ಭಾಗ ಕಟ್ ಮಾಡಿ ಇಲ್ಲಿ ಇಟ್ಟಿರೋದು. The protoxylem, it is towards the center, and the metaxylem, it is away from the center, or it is towards the pericycle, or it is towards the periphery anta kuda helbodu. So this type is called as endar. So the vascular tissue here, it is conjoined, means it is present in same radii. It is collateral. Collateral means xylem is towards the center and phloem is away from the center. Open type. What is open type? Open type means in between xylem and phloem, if there is a presence of cambium tissue, then it is called as open type. Endar condition is a condition where the protoxylem is towards the center and the metaxylem is away from the center. So I'll write this diagram once again here. Just go through the diagram. So first there is a patch of phloem tissue. Patch of phloem tissue. Sieve tubes, companion cells will be there. Phloem tissue. Next to the phloem tissue, we can see the cambium meristematic tissue, which is in a continuous state of cell division. This cambium, remember, it helps in secondary growth. Why it is termed with the word open antandre? Because this cambium or this stem, it is open for secondary growth anta. It is open for secondary growth. Andre the stem, it will undergo next stage, that is the secondary growth. Hence it is called as open. Next is xylem. See, the protoxylem, it is towards the center. This is pith region. Next, the protoxylem, this larger one, it is called as metaxylem, larger vessel with a wide lumen. Next is cambium, and this patch of cells is the phloem. So hence the vascular bundle, it is conjoined. Xylem and phloem, it is present in the same bundle. Collateral, xylem is towards the center and phloem is away from the center. Open type, because in between phloem and xylem there is a patch of cambium. Endar condition, protoxylem is towards the center and metaxylem is away from the center. Now, in between this two vascular bundle, there is a patch of loosely arranged parenchyma cells in between the vascular bundle, and this region is termed as medullary rays. A patch of parenchyma cells or a parenchyma tissue which is present in between the vascular bundle, this region is referred as medullary rays. What is medullary rays means? You can see here also. In between the vascular bundle, you can see a patch of parenchyma cells. This patch of parenchyma cell in between the vascular bundle is the medullary rays. And the central region, it is made up of a large parenchyma cells with a large intercellular space. See here the intercellular space is less. But when it comes towards the center, the intercellular spaces are more. So hence at the center region, it is called as pith region. So this center part of the stem, it is called as pith. In between the vascular bundle, this region is the medullary ray, which is made up of parenchyma cells. So this is about the anatomy of a dicot stem. So the anatomy of a dicot stem, it is mainly differentiated into three regions. The outer region is called as epidermis. The outer region is called as epidermis, which is made up of the compactly arranged barrel shaped parenchyma cells, and certain parenchyma cells, it is modified into stem hair. And also we have to say here the another one, the outer layer of a dicot stem, it has a waxy layer called as cuticle. Cuticle is seen. Cuticle, the waxy layer is present. The outer to the epidermis, there is a waxy layer called as cuticle, and certain epidermal cells, they are modified into the stem hairs called as trichome. Stem hair, or you can also say it as trichome. So this is about the epidermis. Next is cortex. The cortex, it is differentiated into 3 regions. That is, the outer region of the cortex is hypodermis, middle general cortex, and inner endodermis. The hypodermis, it is made up of collenchyma tissue, 2 to 3 layer of collenchyma tissue. Then comes the general cortex, which is made up of loosely arranged parenchyma cells, 2 to 3 layer of loosely arranged parenchyma cells. Next, the inner layer of the cortex is the endodermis. The endodermis, here it is made up of single layer of the barrel shaped parenchyma cell, and this endodermis, it is also called as starch sheath. It is called as starch sheath. Why it is called as starch sheath means there is a deposition of starch granules in this region, hence it is also called as starch sheath. So this up to the endodermis, it is termed as cortex. From hypodermis to endodermis, it is cortex. Next to the cortex is the stellar region. This is the stellar region, entire region. Again, the stellar region, it is differentiated into 3 regions. One is pericycle, next vascular tissue, and central region is pith. So here the pericycle, it is discontinuous. You can see here it is not continuous. Here you can see a patch of sclerenchyma here, then there is no sclerenchyma, then again here patch, here it is again a patch of sclerenchyma. So here it is discontinuous. The pericycle is discontinuous, which is made up of a patch of sclerenchyma tissue. In between the sclerenchyma, we can see a parenchyma cells. So this sclerenchyma, it is seen above the vascular bundle, like a cap-like structure. Hence it is also called as bundle cap. Next is vascular bundle. The vascular bundle means it is consist of xylem and phloem. So the vascular bundle, when we take up with one vascular bundle, the xylem is towards the center and this phloem is away from the center. So hence it is collateral, it is, as it is present in same bundle xylem and phloem, it is conjoint, and it is open type. Why it is open type means in between phloem and xylem we can see a patch of cambium, hence it is open type. Next, the protoxylem is towards the center and metaxylem is away from the center, hence it is endar condition. And in between this two vascular bundle we can see a patch of parenchyma cells, or we can see a parenchymata cells with less intercellular space, that is called as medullary rays. And at the centermost region we can see a large parenchyma cells in the pith region with a large intercellular space, So this is about the dicot stem. The best example for the dicot stem is that is tridax anta ಹೇಳ್ತೀವಿ, tridax, or you can give the best example sunflower, etc. So this is about the anatomy of dicot stem. So in the next class we'll start with the monocot stem. Thank you.